Olivia Beaben, and I'm going to be talking about human eye optics and the optical corrections. So, the human eye optics is the study of light, which is one of the oldest branches in physics. It includes topics as microscope, telescopes, vision, and color, which all have applications in life science. The eye is structured roughly square, approximately 2.4 centimeters in diameter, and the light enters the eye through the cornea, which is a transparent layer in the outer layer of the eyeball. The light is being focused by the lens system into an inverted image at the photosensitive retina, which covers the back surface of the eye, which provides nerve impulses that convey information to the brain. The light turning into an image at the retina is produced the curved surface of the cornea and by the crystalline lens inside the eye. The focusing of the cornea is fixed, however, the crystalline lens, which allow the eye to view objects over a wide range of distances. The front of the lens is the iris, which controls the size of the pupil, which also can be the entrance aperture into the eye. The aperture of the eye ranges from 2 to 8 millimeters, and it's depending on the intensity of the light. The cavity of the eye is filled with two types of fluid, both of which have the refractive index same as water. At the front side between the lens and the cornea is filled with watery fluid called aqueous humor, and the space between the lens and the retina is filled with the gelatinous vitreous humor. The focusing of the eye is controlled by the ciliary muscle, which can change the thickness and the curvature of the lens. This process is called accommodation. When the ciliary muscle is relaxed, the crystalline lens is fairly flat and the focus of power of the eye is at minimum. This happens because the light from a distant object is parallel, then the eye is more relaxed. And the distant case can approximately be 6 meters and beyond. Opposite viewing of closer objects requires a greater focus power. The light from a nearby object is divergent as it enters the eye, and that's why it's more focused strongly to form an image at the retina. However, there is a limit to focusing power of the crystalline lens. With maximum contraction of the ciliary muscle, a normal eye of a young adult can be focusing on objects about 15 centimeters from the eye. So if we take a little bit of a closer look at the retina of the eye. The retina consists of photocellular cells in contact with the complex networks of neurons and nerve fibers, which are connected to the brain via the optic nerve. The light that is absorbed by the photoreceptors produces nerve impulses that travel along the neutral network, so the light must pass through all cell layers before it reaches the photoreceptor. And there are two types of photoreceptors in the retina: cones and rods. Cones are responsible for the color vision in daylight, and rods provides vision in dim light. And near the center of the retina is a small depression about 0.3 millimeters in diameter, which is called fovea. It consists of cones packed closely together, and one cone is about 0.002 millimeters in diameter. And the most detailed vision is obtained from that part of image is projected on the fovea. In the fovea, each cone has its own path to the optic nerve. This allows perception of details into the fovea. And then vision and the nervous system. The vision cannot be explained entirely by the physical optics of the eye, because there are many more photoreceptors in the retina than fibers in the optic nerve. That's why we know that the image projected on the retina is not simply transmitted point by point to the brain. A large amount of signaling processing occurs in the neutral network of the retina before the signals are transmitted to the brain. The neutral network works in a way that it decides which aspects of the image are most important and stresses the transmittal to those features. So in these pictures, I chose the guy who is driving, and the eye is then seeing the cars as the most important part of the image. So it's sending stress transmissions to those features, like the car, so maybe he needs to stop the car or try to look what's happening around the environment there. And the effect in the vision. So within the eye, there are three types of defects that has to do with the focusing system. First defect is myopia, and the second one is hyperopia, and the third is astigmatism. And we're only going to be talking about the myopia and the hyperopia. So when the normal eye is relaxed and focuses, the parallel light comes directly to the retina. However, in myopia eye, the lens system focuses the parallel light in front of the retina. So it doesn't come to the retina, it stops right before. This leads to misfocusing that is caused by elongating eyeball or exclusive curvature of the cornea. And in hypertomia, the problem is opposite. The parallel light go way back from the retina. So it goes to the retina and then even further on. And this is caused by the eyeball is shorter than normal. And the hyperopic eye can see objects far away and not near. As for optical corrections for this, so the optical corrections for this would be a lens. So the myopia eye will, for example, probably be able to see objects 2 meters from the eye, and the light that is farther away from this will focus in front of the retina. And the purpose for this lens is to make a parallel light appear from distances farther away from 2 meters. With this kind of correction, this kind of corrective lens, the eye can focus on light way further. And the optical corrections for hyperopia will be to fix a lens so that the eye can focus properly on close objects, where the idea will be to make the eye believe that the light comes from a distant place than there actually is from the close. So the purpose of the lenses are either way to make a parallel light to stop at the retina, not go before it and go after it. And here we can see the pictures of myopia corrections. You can see it stops right at the retina, and myopia without the correction will stop before. This was my presentation about human eye optics and optical